welcome to the Talking Crop podcast. I'm Kathryn Seebruck, commercial agriculture educator with University of Illinois Extension, serving Daviess-Stephenson and Winnebago counties. Talking Crop is a seasonal crop production podcast with episodes occurring every other week during the growing season between the months of May and September. In each episode, I bring on a guest speaker to discuss topics related to their areas of expertise. Today's episode is the final of this first season of Talking Crop, and in it, I am joined by Dr. Andrew Margenot, Associate Professor of Soil Science in the Department of Crop Sciences at University of Illinois Urbana-Champaign. Andrew is also the director of both the Morrow plots and the Alma Mater plots at U of I. He and I discussed the Illinois Centennial Soil Archive project, as well as his work to update the phosphorus and potassium recommendations in the Illinois Agronomy Handbook. The story behind the Soil Archive project is fascinating as it involves century-old soil samples found in an old building on campus and will lead into a really interesting project that will uncover valuable information about Illinois soils across the entire state. And producers around the state are going to hugely benefit from updated phosphorus and potassium recommendations and may even get to play a role in the research that will result in these updates. So listen on to find out more. An exciting update about the podcast as a whole is that we are adding it to the Illinois Extension website. So, if you're listening after the fact, you may be a little confused to hear me talk about adding links to the video description as the podcast originated on YouTube. So just a little FYI there. As I mentioned before, this episode wraps up the first season of Talking Crop. I'm really appreciative of the guest speakers who joined me this season and for all the great conversations that we've had. And I hope you, the listeners, have also enjoyed it and have gotten something valuable from these conversations as well. I'm already looking forward to next season, which will come back around in May of 2025. Without further ado, please enjoy this final episode of the first season of Talking Crop, talking 100-plus-year-old soil samples and their implications for Illinois with Dr. Andrew Margenot. Andrew, thank you so much for joining me today for the Talking Crop podcast. I appreciate you taking the time to be here.
Of course, Kathryn. Thanks for having me.
Sure. So the two main things that I want to talk to you about today are your two of your big projects. So the first being the Illinois Centennial Soil Archive project, and the second being your work on updating the phosphorus and potassium recommendations for Illinois. And I wanted to start with the Soil Archive project. I know I've heard quite a bit about this, and I've seen some things, floating around here and there, and it's a really interesting project. So could you start by just explaining the background of this project and what all it entails?
I'd be happy to. So the project, as the name might imply, has to do with a rare collection of soil samples as far back as the 1800s. The oldest sample we have is 1861, and the most recent samples are from 2 years ago. So this is a soil archive that was developed from the original soil sampling of the Illinois State Survey. So basically, we know that we've got soil maps of any given farmland in the contiguous lower 48 states. That was a long effort that began in the 1800s, like in our state, with scientists from the land grants going around describing soils, where they dig a soil pit, and they look at every single layer in that pit, these different horizons, as we call them. And they describe soils, they give them a certain classification, and over time, this led to the development of these maps, soil type maps. Well, it turns out that the samples for the Illinois survey were kept on campus and stored. It seems like they exchanged different hands and different storage places. They used to be at one point in Davenport Hall on the main quad. Then they were put into the tunnels beneath Turner Hall, and they ended up at a dilapidated pole barn on the South Farms, where we rediscovered them in 2018. So there's over 8,000 samples, and these correspond to 460 locations across the state, because keep in mind, each location has multiple depths and thus multiple soil samples. This is exciting because the samples go back with pretty high coverage of the state through the 1910s and 1920s, through the whole 20th century. So in sum, this is what we believe to be the world's largest in terms of area coverage, but also the oldest archive it's a huge resource for us to understand soils across the state.
Okay, and so you were mentioning that they were originally collected for to develop those soil maps of the state. So are these the same soil maps that you see, for example, like on the Web Soil Survey online?
Exactly right. So the soils that were taken from this effort were the basis for the PED-ON characterizations. A PED-ON is simply a soil individual. And so these characterizations of soil properties, et cetera, and where you find certain kinds of soils were then used for the SURGO as well as the NCSS data set, simply the data set from which the NRCS pulls the properties like CEC or clay content.
Okay. And so these were obviously taken a long time ago. And I know we'll talk a little bit about how you're getting basically these repeat samples now. So way back when, do you have any information on how those samples were taken in terms of, I think something your team might struggle with is getting permission to go back and resample. So back in the day, do you know if they had any issues with that or if it was kind of mandated that they were allowed to do so?
That's a great question. So here it might help to think about the two phases of soil survey. Basically, it's before and after NRCS. And really,
Mm,
NRCS was at one time called the Soil Conservation Service, SCS. So
sure.
it's really pre-SCS / pre-NRCS or post. It began with states. It was first a state endeavor to map soils. The first soil survey ever was in 1899 in Illinois, in Adams County specifically. That was the first documented county effort. Now, there were efforts before that, hence our 1861 sample. And then after, I think about the 1940s, don't quote me on that, we have the feds helping cooperate. So this was called cooperative soil survey. For the latter, I believe it was a combination of networks and also as part of a federal effort, then the permissions could be gotten. The idea was that mapping a resource was part of federal rights to get onto private property. Now, the pre, or let's say the phase one pre-NRCS, I don't know. That's a good question. My sense was that it was a lot of knocking on doors and asking
Hmm.
for permission to sample. This is also at a time, Kathryn, when soil science was in its infancy, and it was a big deal to get a researcher from the U of I come knocking to help understand soil types. We have the notebooks by Dr. Hopkins, the first soil scientist, in my office right here in front of me. These are the notebooks in that pre-NRCS stage. They're in Quillink and they've got dates. One actually says September 4th, 1901. These are thousands of pages of entries, and it seems like from his notes that he was knocking on farmhouse doors because we have notes like by the red farmhouse, 200 feet away from its oak tree in the front lawn, those kinds of things. It sounds like it was based on personal interactions. So to back up today, we do have the challenge of finding permission from the landowner to sample. And we are able to find who owns land. That's public record. And there's apps like OnX for hunting, where they pull into the public database from the tax records who owns the property. There's two issues there. One is that a lot of land is now owned by investment firms. Not a lot, but more and more land is owned not by a person, but by a corporation or LLC. And that's a dead end for us. The second is that even if we have a name, we would like a way to get in touch with them directly, like a cell phone. And that proves
Mm-hmm.
to be the bottleneck in finding or asking for, rather, the permission to resample.
Okay. And so, like I said at the beginning, I've heard quite a bit about this project. And so, are you guys putting in a lot of effort then to try to get the word out so that you can hopefully find those folks and subsequently, like you said, their contact information?
We sure are. Here, we've tried different approaches, describing this effort on social media, like Twitter, uh, through Farm Bureau. We've given a lot of talks at the county level Farm Bureau offices at field days where we had talked about this project. And so it's been pretty successful. I want to point out that a lot of alumni, especially, have been fantastic in reaching out and giving permission. And because of their efforts, we have either sampled or have gained permission to sample this fall roughly 130 of the 460 sites. So that's a little bit more than 1/3 of the way there. That's a big deal. And
Mm-hmm.
so for the remaining 2/3, I think that's where, well, we've picked all the low-hanging fruit. And it might be folks who have not heard about the effort, or might be landlords who don't live in the state and thus haven't heard about the effort.
Sure. And so part of your guys' reasoning for doing this, at least what it states in the kind of the promotional materials, I'll call them, that you've put out, are that reasoning is to kind of look at how soils have changed in the state between when these original samples were taken and now. And so you said you have these notes and records from way back when. And my question then is, were the things that you guys intend on sampling for, I guess, testing in these samples, were those same things tested for back then, or are you going to have to sample those original samples yourselves and then the subsequent repeat samples?
A mix of both is the short
Okay,
answer. So
sure.
it's a great question. I will back up here and give a bit more on why we want to resample these, just to give
Mm-hmm.
folks some more rationale. So um, the samples that we have already are a treasure trove because we have samples that precede a lot of important innovations in ag technologies like fertilizers. We
Mm-hmm.
used rock phosphate as a state by the 1910s, but really it was a small total amount of phosphorus being applied as rock phosphate. Potash was rare. It was more common by the 1930s and 40s. But really, we don't see large-scale application of NP and K until post-World War II. And for example, MAP or DAP weren't really available at scale until about the 1960s when we have USDA beginning to keep records on sales for the reason that they're now exploding. So the soils that we have, for example, that predate the wide-scale use of NPK, let us test questions about what was the native stock of P and K in soils. Because we have every layer going all the way down, we can come up with a pound per acre of the total amount, what we call a stock of a nutrient, pounds per acre. These are not Bray or Malik, it's the total concentration. So there's value in things like that. We can look at the emergence of contaminants like lead or microplastics when they first become detectable in soils. There's all kinds of questions. Think about weed herbicide resistant. We can look at weeds at the seeds of weeds in
Mm-hmm.
the archive jars, which are unground soils, and then pick them out and sequence them. So a lot of potential here. Now, we want to also resample soils. And in the resampling, we can understand by comparing the original soil property when it was sampled at that time with our reanalysis of a newly sampled soil, what has changed. Your question then is, well, have some of the soils that were taken originally been measured for a certain property of interest? And the answer is sometimes, but not always. When we have a physical sample from the past, we can reanalyze it. So a good example here is P and K. We have interest in mapping P and K levels across the state before the advent of inputs of MAP, DAP, and potash. And for this, there weren't a lot of samples. There just were not a whole lot that were sampled for total P and K pre-1940. So for those, we can re-measure that, or let's just say measure on the archive sample. And it does present an issue of how these jars were stored because there is no gas phase for P and K. There is for carbon, for nitrogen, and for sulfur. And because of how these soils are stored, they're air dry, which is important, it's critical, but they weren't exactly temperature controlled, being in a pole barn for at least 20 years. We may have lost some of the carbon and or sulfur through gaseous phases over the past, centuries or decades of storage. Now, we do have some samples that were measured for C, N, and S, those three elements that have a gas phase, and we can reanalyze that sample to look at the change as a function of storage. So there's two kinds of measurements. Samples that were originally measured at the time of sampling look at the susceptible to loss nutrients, C, N, and S. By remeasuring that decrease that we would maybe see is due to how it was stored. And then we can correct any fresh first-time analyses we do on the original stored samples. So that would be how to back correct for those artifacts of storage. And then on the new samples, we run everything as is. So we have pretty high confidence Now, that would be for fertility. For things like properties of CEC or pH or clay, those are agnostic to storage. And so we can remeasure those in a sample from 1860, no problem. The third kind of analysis that we're planning to do that's a bit more contentious would be soil health tests. And
Mm.
as you might imagine, these are largely emphasizing the biology of soils. We have done some preliminary analyses of the DNA, so looking at the microbial community composition using DNA sequencing and also enzyme activities. Soils are full of enzymes, just like our stomachs, to digest, in their case, organic matter. And these are made by plants, by the roots specifically, and by microbes as a way to digest organic matter and then liberate nutrients like nitrate or phosphate. Well, these enzymes are extracellular. They're not in living cells. So when we air dry soils, the enzymes persist. They simply bind to the clay surface. And then when you re-wet the soil, they are activated. And for the DNA side of things, we can extract that because DNA is very stable bound to clays. Long story short, we have evidence from doing some initial tests with the Morrow plot samples from 1904 that we can resurrect what's called the fossil microbiome. I think fossil
Hmm.
is a bit of a dramatic word here, but it's a term inherited from all these studies in paleobiology where they, like in
Hmm.
Jurassic Park with the mosquito that's in the amber, analogous to that. That's not actually possible, but analogous to that, extracting DNA from these older samples. So that's a long way of saying that we're able to, I think, have with decent confidence understand what things were like when they originally sampled these soils, and also by looking at the resampled soils, how things have changed.
That's really fascinating, and you answered a lot of questions that I didn't even think I had initially. And so I'm jumping ahead a little bit here. I know we'll specifically get to that updating of the phosphorus and potassium recommendations, but something you said made me question. So was your team and potentially others, I guess, depending on who's involved. Was that update of the phosphorus and potassium recommendations, was that already being thought about and implemented before these soil samples were found, or were the finding of these soil samples kind of the impetus for something like that? Since once you found them, you knew you had them on hand and could kind of look at how we can update them from back then to now.
Yeah, well, they were kind of contemporary. They both emerged within the same 2-year time span. And when I say both emerged, the discovery was in August of 2018. I remember
Okay.
because I was so like thunderstruck that I spent the whole day out there with my headlamp. And then at the same time, we were trying to develop what would become this larger scale project and updating the state agronomy handbook recommendations on P and K, but they certainly fed off the other. So one key aspect of how we think about P and K is the critical value. This is a number that you want to be above on your soil test. So for
Mm-hmm.
phosphorus, it's typically 40 to 50 pounds of Bray P per acre. You want to be above that number. Well, those values are in part based on soil supply power, basically how much natively occurring P and K is there in your soil. And that assumption requires, I think, really good data measuring P and K stocks across the state. And that then crosses over to the archive where, oh my gosh, we can now actually quantify what the native amounts of P and K are.
Mm-hmm. Sure. Yeah, because I know we've kind of worked tangentially on a project together with some of my other extension colleagues looking at that chapter of the Illinois Agronomy Handbook. And you mentioned how there's a map in that chapter that has this kind of general idea of the different areas of Illinois and what their inherent P and K values are. And I know you've mentioned a couple times how that definitely needs to be updated. And so did that information, do you know if that information came from this sampling project, or was it just kind of a generalization from some other source that maybe someone back in the day kind of had on hand?
I think it's the latter. And I say that because so in chapter 8 of the agronomy handbook, there's a figure that shows the P supply power, also called
Mm-hmm.
subsoil supply. And that map is based on the loess thickness and on the loess age of the state. So loess, LOESS is the parent material that blankets the state. Soils were born out of the parent material, and so that would be loess for us largely. And think of this as the fine silty stuff that was blown in post-glaciation.
Hmm.
So there's a lot of this stuff across Illinois. It's thicker on the west, more or less, and it more or less thins out as you go from a northwest to a southeast transect, by and large. Okay, so the map that was used for the agronomy handbook said if you have deeper and or younger loess, so as you go more north state, you get younger loess because the glaciation stopped later, then you're going to
Hmm.
have more native phosphorus. Well, those are based on maps that we believe are from the 1940s, and we found some other maps in the 1960s on the loess age and thickness. So the basis for the supply power of phosphorus in our subsoils that then informs in part the critical values, that was based on a geological concept of the
Okay.
loess thickness. It's not related to any measurements that we can tell of the actual P content of these subsoils.
Sure. Okay. So it sounds like the project that we'll talk about in just a couple minutes here is going to be a much more thorough kind of investigation into what those supplying powers or what those values actually are. But
Yes.
like I said, I keep fast forwarding to that portion. So before we do that, I just wanted to talk about a couple other things with that Centennial Soil Archive project. So you mentioned how there's hundreds of samples and many locations. I know you said you've made some kind of breakthroughs in getting those numbers up in the resampling. But obviously you're not completely finished yet, unfortunately. So where would someone find information about, I guess, this project as a whole, but then information about where these remaining samples are at?
I'm happy to discuss that. So yes, we have over 300 samples left to take across the states. And I should say site, because at each site we take a deep soil core down to about 3 foot depth. Where can folks find more info? Well, I think the key is the map. For this, if you go to our lab website, which is my last name, Margenot, Margenot, cropsciences.illinois.edu. It's a bit long, sorry, but if you just Google also U of I, Margenot, you'll find it. On the lab website, there's a tab at the very top that says Illinois Soil Archive. And that page contains the background information on what is this, why are we doing this project, and there's a link to the Google map. Now we have the link in two forms. One is as a QR code, meaning on your smartphone, if you scan it with your photo app, it'll automatically pull up the Google Maps on your phone. I personally feel like the Google Map on the desktop or on the laptop is easier to navigate, and we have a hyperlink in green text for that. So that will also plug you into the Google Map format, and it looks like a Google Map. The difference is that we've got 460 pins across the state of Illinois, and each pin corresponds to a original sampling location. How to use this map? Well, there's three kinds of pins. There's yellow pins, there's green pins, and then there's a red check mark pin. As you might imagine, the red check mark is a site that we have already sampled. So huge thanks to those landlords, operators slash owners for giving us that permission. Then there's the green pins, which are sites that, again, big thank you, we have been granted permission to resample. And then we have, of course, the most of the yellow pins, which are sites that have yet to be identified for a landowner name and contact, which we would then be using to request permission to sample in the coming 1 to 2 years. Overall, if you look at this map, we've got some gaps in the south, especially. So once we're south of Effingham type area, really south of 70, we've got a pretty large gap until we hit the very southern tip, like Alexander County. And then up north, up in the Driftless, for example, we've got some pretty big gaps. And then out in the south west area. You can zoom in to these pinpoints, look at fine scale resolution. It is a Google map. If you click a pin, you'll find that you get what's called the PEDON ID. So again, Pedon is a soil individual. It's a example soil that's been sampled, and that tells you information like the exact GPS coordinates and the year of sampling. I just clicked on one right here. That is Montgomery County, 1960. Uh,
Hmm.
just clicked one up in Kankakee from 1930. So it gives you a flavor for how old these samples are in terms of when they were last taken.
Okay, perfect. Yeah, that's really good information to have. And I've perused that map, and I would agree it's much easier to navigate on the desktop. So I would recommend that as well. And then you mentioned how you take a 3 foot sample. Would the landlord or landowner or tenant, whoever, would they get the information that you guys obtain via these samples?
Absolutely. So a bit more on how we sample. Once we have a contact name and phone number, we make a first call just to, ask if we can get permission. When we're in the same season of sampling. So for example, if we get permission to sample a site in December, that's a bit too late for us to go out there. So that would likely be a pre-plant sample as things thaw out before crops are being put in. We would give a 1 or 2 month out heads up of, hey, as a reminder, if the offer still stands, we'd like to go out there. Then, roughly the week of the sampling, we design a circuit of what sites can we hit. As you might imagine, we have a lot of equipment on a trailer to haul out there, so we have to design circuits,
Hmm.
and we would give a heads up phone call that week. We get out there, we take 4 to 6 different cores. They're about 3 foot deep. We have not punctured any drainage tiles yet, and we don't think that we will because we work with the landowner operator if they have concerns about that to where can we sample to avoid a tile.
Sure.
And the depth that we go to will be more shallow. If there's any concerns, we simply don't sink the probe down to 3 foot. So then those samples go back to campus. We then process them every 6 inches, all the way down to 3 foot. We take a separate section, and that gets analyzed for a lot of things. These are soil properties like clay content, pH, CEC. We look at soil fertility, the total and the available of everything, macros and micros. And third type of analysis is soil health, the kitchen sink of all of them. It's about $4,000 to $5,000 worth of analyses that we perform. All that data goes right back to the owner operator. And that also is not just a data dump of a lot of numbers. We provide a report that interprets and visualizes with graphs and figures exactly what it looks like. And we also compare it to the original sample. So then the owner can see, if grandpa farmed this in 1940 and someone took a sample, how have things changed? All this data is kept on private servers at the U of I, so only our team has access to it. And the exact sample location is never revealed so that it cannot be traced to that property.
That sounds like a lot of really great information to get and for free. And on top of the fact that you get that extra added bonus of, like you said, looking at that historic information. So hopefully you guys can get those numbers up and get this finished in due time. It's
I
a
hope
really
so.
great project. So jumping back then to what I was talking about before, I got a little overzealous and jumped ahead to our next topic. But your project, like I've mentioned, at updating the Illinois phosphorus and potassium recommendations. So again, I know we've talked about it and touched on it, but can you briefly go over the background of this project as well?
Sure thing. So this is a companion or a sibling project, as you've mentioned, where we're trying to update specifically Chapter 8 of the State Agronomy Handbook, the recommendations on P and K. And we're not just trying to verify if the current recs are still valid, we're trying to add levels of nuance and complexity. And there's at least 4 different levels of complexity, but I'll begin first on what the chapter updates are. In Chapter 8, we have what's called a critical soil test value, also called the threshold value. Some folks call them CSTVs. It's all the same thing. It's the idea that if you're underneath this number on the soil test, there's likely a pretty good chance of seeing a yield response to adding more of that nutrient. So we don't do this for nitrogen, right? That's based on a different paradigm. We don't soil test for N rates because it rains a lot. That works out west where the rain doesn't push out the nitrate. But out here where it rains, we soil test for P and K. And we then want to know, hey, if I spread MAP or DAB or potash, is it going to be worth it in terms of a yield response? Those critical values then, they vary by region due to the supply power concept for P or due to CEC for K. So for K, the paradigm is that if you're on sand, like up in some parts of Kankakee, you can't really build K levels because CEC is so low. And if you're in a low versus high K, then your critical value for K is also different. But the power of this stuff, which was first developed in the 1950s, is that it gave farmers at the time the first ever quantitative way to have a higher confidence approach to adding nutrients. Until then, it was kind of guessing. Now we have a way to measure what's in the soil for the crop. Now, the issue, I think, is that these values have not been updated since the 1950s, as far as we can tell. They've been modified based upon what our neighboring states do, but a lot of these recs are, number one, pretty coarse. We only have 3 different regions for the P supply power concept. And also, that concept has not actually been tested. We don't really know. Is it valid to adjust what your critical grade p-value is based upon whether you're medium, low, or high supply power? That's an assumption being made with no data. Other details that we have to address is that no one actually measures Bray. I know that we still talk in Bray P, also called the P1 test. It's all the same. But all commercial labs are now running soils for the Melic 3P test because they also can measure K. So we've stopped using Bray P and ammonium acetate K. We now, if you get numbers that are saying P1, they're measuring Melic 3P and they're back calculating by a correlation what the Bray P value is. And they're roughly comparable. They're about 35% difference in what the value is. So that's an example of how it's time to update to what the commercial labs are giving you back on your tests. Other issues include things like the depth of sampling. So what we're adding as a new expansion is we want to go to a second depth, not just the top 7 inches, but the 7 to 14 inch depth, because we know that crop roots can forage P and K beyond the top 7. And here's a good example. In the south part of Illinois, we have a lot of fragipans, clay pans, and P and K will move, especially K, downward. They'll leach out of the top 7 inch depth from which we sample, but they'll sit on top of that clay pan at say 7 to 14. So the corn or soybean is seeing plenty of K. It's doing just fine, but your soil tests at the top 7 would miss that underlying 7 to 14 inch buildup of P and K. And you would conclude incorrectly that you got to spread P and K when in fact the crop is doing just fine. That's an example of how we should go a little bit deeper, or at least let's test whether going deeper on certain soil types improves the prediction of a crop yield response. The second major new thing that we're adding is looking at soil type. So we want to make critical value recommendations, not by broad regions, 2 or 3, 2 for K and 3 for P, but rather by soil types. As you pointed out, we have these maps from Surgo, soil type maps for every single farm across this country. Why not link the P and K critical values to the soil types? This will enable VRT. It just enables more precision management of our nutrients. And for this, we are doing on-farm trials broken out by specific soil types so that we can try to capture the 57 major soil associations. So soil associations are not soil types like drummer or verdin or Sisney. Instead, we get associations like Ava Sisney or Flanagan drummer Catlin, which are sibling soils that occur on the same part of the landscape. You often find a farm will have, Flanagan and drummer, Sisney and Ava. That's why we're breaking this out by those associations. So there's 57 of those across the state that we want to try to link the critical values to. The third change that we're doing, well, actually the fourth, excuse me, is the economics of P and K. So right now, the critical value that you want to be above this, so you should be building the test value, that's based on yield response. But as all farmers know, maximizing yield does not necessarily maximize net profit. And in some cases, you can lose net profit by increasing yield because the extra P and K won't pay for itself. And we've seen some pretty rough high price years for P and K. back in post-COVID, and we're seeing not great grain price years this season.
Hmm.
There might be years where there might be a yield drag of a couple bushels per acre by not spreading P and K, but it'll still be more profitable because the P and K won't pay for itself. Clearly, there's a point where we have to put it on because we're taking off P and K with every bushel harvested. But there needs to be, I think, some information on the economics of P and K. Does it make sense to apply from a net dollar per acre return? So that's the final novel aspect of our updates. These will all be part of an online tool, just like the MRTN, where you can click, input your soil type, your soil test, even the prices of inputs and the grain to estimate what might be a good input rate.
That's awesome. That sounds like it'll be at the end a really invaluable resource that you guys are going to be putting together when this is all said and done. So you mentioned that the study is being done via on-farm research. And so what then, I guess, what do the treatments look like on this on-farm research? And is it something that you're still looking for participation on, or have all those kind of slots been filled, so to speak?
Great question. The answer to the second one is no. Slots are still very much open. I want to emphasize that we're happy to talk to folks. Yeah, this is a big effort. So we need hundreds of site years. A site year is the total number of locations and years. So if you have 5 sites here, it could be you have the same farm for 5 years. That's 5 site years, or 5 farms in 1 year. That's 5 site years. We think
Mm-hmm.
we need at least north of 360 site years. That's
Okay.
quite a bit. So for this, what is involved exactly? Well, we try to find farmers who want to host us and are willing to put up with our nagging, where what they provide to us is a file, a shape file of their field of interest. And we're taking fields that have any soil test level of P and K. We, in fact, on purpose, want from very deficient to medium to adequate to very high levels. We need a full spectrum to make these recommendations correctly. So don't think that you have to have a P or K deficient field to participate. We then look at the shapefile, and our data scientist makes the prescription maps where we would be proposing the omission trials. So the way to come up with a critical value is that you compare the yield without the nutrient versus the yield with the nutrient. Soil tests are taken on those two plots right before the application, and then we correlate the soil test value with that relative yield. Relative because it's the yield without versus the yield with. That's one data point. We need 400 of them to make
Mm-hmm.
decent confidence estimates. So what we ask from the farmers is that if you give us the shapefile of what you think might be a good candidate field, we do all the work of identifying by soil type where these could go. These plots are about 120 foot by 120 foot, and we've got 8 of those per field. So 4 pairs of with and without, and that would be for a given nutrient, just for P or just for K. In some fields, if they're big enough, we do both P and K trials, so 16 plots total. We take care of all the soil sampling. We work with the applicator, be it the farmer or be it a co-op, whoever, to make a single prescription map so that in one single movement across the field, in one event, both the rest of the field gets applied and then these specific plots. And then the other ask that we make of the farmer is the yield map off the combine. From that, we can come up with these critical values based on the relative yield. So that's what's involved. We're happy to meet with someone to visit by phone first to just talk them through. And we're looking for either fall applied or spring applied sites. So there's still time for both for the 2025 season. And there's no commitment. If you do 1 year, that can be a one and done. If you want to keep the same field going for all 4 years left, also a possibility. And finally, we can do more than one field per farmer.
Okay. That sounds like a uh pretty good deal in terms of what all is being done, not too intensive. Does the farmer or landowner, again, whoever you're working with here directly, get like any sort of compensation for being involved in this?
Yes, they get what we call the nuisance fee, excuse me. So we pay the farmer $1,000 per field for a single nutrient trial. And if you're doing P and K in the same field, then that's 1,500 that we compensate them. We don't think that there's going to be much yield drag, right? Unless we've got
Hmm.
very, very low P or K values, like we're talking, single digit Bray P. Then there might be a yield drag. The payment that we have would cover the loss of profit in these 160-foot square plots. So we're talking pretty minor yield drags. If we see a larger one that is an economic hit for the acreage that we have, which is about 1 or 2 acres per field, we are happy to talk on how to compensate them. So we have a flat nuisance fee of 1,000 to 1,500 per field, depending on how many P and or K trials in that field. And again, the ask is the shapefile on the front end, and then the yield off the combine on the back end.
Okay. And if someone determines that they might want to be involved in this, how would they go about getting in contact with you and your team?
Sure thing. Before I give that information, one last note. I know
Sure.
some folks are maybe hesitating to spread P and K this fall because of the pretty low prices on grain. We don't have to apply, or the whole field does not have to be applied.
Hmm.
So it is possible to just have the P and K applied on these 160-foot plots. So
Okay.
it doesn't mean that the whole thing has to get P and K if someone is thinking about not spreading. How to get in touch? Well, the best way is my email. That is my last name, Margenot, Margenot, at illinois.edu. If you again Google my last name and U of I, you'll find the lab website and my contact there. Then myself and our data scientist, Ezra, would work with you to check out the fields, meet with this person about whether it's a good fit, and if so, we'd move forward with all of the application prescription maps.
Great. Perfect. And that information and any other links or anything of that nature that we've talked about will be included in the video description for those listening. So that'll be easily accessible for you guys. So, Andrew, thank you again so much for joining me today. This was chock full of really great information that I know a lot of people are interested in hearing, and hopefully we can get some either some new samples for you guys or maybe some farmer participation in your project out of this. Best case scenario, right?
Yeah.
So again, thank you so much. I really appreciate it.
Thanks, Kathryn, and thanks for having me and for letting me yap a little bit about these projects. I think I personally have lost sleep over both of them, but the archive one, when you hold a sample from 1861 in your hands, that's pretty mind-blowing. And we can learn a lot about our soils to help from applied to more fundamental questions on soils and their fertility for the state. So any help that we can get on either of these would go a long way. Thanks for having me.